Good morning, how do you do?
Very good, thank you.
Nice to see you, nice to meet you.
Likewise, likewise.
My name is Kurtov German.
Nice to meet you.
Nice to meet you. And this is Dmitry. And my English is so bad, so I will speak Russian if you don't mind. And Dmitry will speak English. Because he is just... it was his idea to contact you, to communicate with Zoom. And if you don't mind, I just push the record button. Okay? Just a second. Just a second.
Recording in progress.
Recording is in progress. So I just start speak Russian and okay. Всем добрый вечер.
Well, as you've introduced, my name is Jeff Meldrum. I'm a professor of anatomy and anthropology here at Idaho State University, where I teach human gross anatomy in graduate health professions programs. So I'm very familiar with the human body, its form and function. My research focuses on the evolution of human bipedalism, that habit of walking on two legs, and the adaptations that are associated with that. So that involves research into the hominin fossil record, looking at the skeletal remains, looking at fossilized trace evidence, the footprint record of hominin bipedalism, as well as studies of living humans and non-human primates, the great apes particularly, our closest kin, in order to better understand our particular adaptations. So it's that interest, particularly that focus on walking on two legs, bipedalism, which drew my attention to the Sasquatch question. And of course, in order to establish a context for a relic hominoid here in North America, the reported potential existence of other relic hominoids has taken on an important interest.
Dr. Meldrum, how did it all begin for you? What exactly brought you to the relic hominoid research?
Okay. Well, as I said, I've long had an interest in human evolution and the hominin fossil record. Even as a youngster, I was very interested in paleontology and natural history, and particularly primate behavior. And as a youngster of about 11 years, Roger Patterson, who is famous for his role in capturing a film clip, a 60-second film clip from Northern California, alleging to portray a Bigfoot, a California Bigfoot, if you will. He was displaying his show, his film rather, within the context of a documentary film in various public venues. And one of the first was where I was living in Spokane, Washington, in northwestern United States. And so I was in attendance. I convinced my father to take me and my younger brother to see this film. And it obviously made a lasting impression. And that interest waxed and waned over the years, but eventually my career aspirations took me in the direction of physical anthropology and anatomy. And so in 1996, a number of circumstances sort of aligned that put me in a position to examine a long line of footprints, very clear 15-inch footprints in wet, very wet, muddy soil. There was no question as to interpreting. They either were a hoax, a very sophisticated, very elaborate hoax, or they were the footprints of a Sasquatch. And by this time I was quite familiar with the distinguishing characteristics of both human and non-human primate feet and footprints, and so both recent and fossil. And so that experience, that wherewithal, put me in a position to, I think, draw a rational conclusion, and there was no escaping that these were authentic, these were real. And that was in 1996. Now, several decades later, I have over 300 footprint casts in my laboratory, primarily here from North America, but also examples of others, including a pair from Russia, and other examples of the almas, orang pendek, the yowie of Australia, which is still a bit of a puzzle. Footprints aren't nearly as convincing from there as elsewhere. So that kind of brings it up to where we are today.
As a primate foot anatomy expert, what do you find special about the Sasquatch foot and how it is evident from the footprints that you have examined? Why do you think they cannot have been hoaxed?
Right. Well, they were extremely fresh. The weather conditions were such, it was in early spring, well, actually late winter, February, still snow just barely melting back. The soils were very saturated, very wet. It was overcast, drizzling rain. So the conditions were such that the details that I was observing, the details of skin texture even, indicated that they could only be several hours old, a half a day perhaps. And so the freshness was very telling. There were all the signs of animation. There was variation from footprint to footprint as the foot. Remember, a footprint is not a static mold or impression of a foot, but it is instead the dynamic record of the foot's interaction with the soil, with the substrate. And so those features were quite evident. There were slide-ins, there were indications of toes extended, toes flexed tightly, gripping the mud and sliding with mud extruding up between the toes. There were tension cracks and pressure ridges within the foot. There was all the indication that these were imprinted rather than stamped. A stamped artificial footprint creates an impact crater with a raised lip. But in this case, you could see it was indented, it was impressed into the soil. So features like that, I mean, there were, in order to have fabricated such an example of multiple footprints, I mean, we're talking 35, 45 individual footprints, would have required multiple sets of artificial feet because of the clear animation and dynamics in the action of the toes and the flexibility, the articulation of the midfoot, producing pressure ridges and such. So I was there, I was literally kind of scratching my head because at the time I was very skeptical of this person. I'd been given lots of advice from others about his character and his reliability, which were in hindsight now, obviously were baseless, or largely baseless, at least in the context of this instance, absolutely baseless. So they, these another in a more general terms, these footprints were very representative of what has by now emerged as the model of or profile of the Sasquatch footprint, which distinguishes it from a human. These are not simply enlarged human footprints. They differ in a number of ways, including they are flat and indicate midfoot flexibility, as indicated by the pressure ridges and a greater degree of supination, pronation, the twisting of the forefoot relative to the hindfoot. They are broader for their length, which one would expect for an animal of this dimensions, because volume increases to the cube of linear dimensions, whereas surface area only to the square. So for increasing body mass, there has to be a disproportionate increase in the surface area, or other anatomical or behavioral modifications, like walking on a compliant limb, for example, with flexed joints, flexed knees and hips, which is very characteristically described by eyewitnesses who see a Sasquatch walk. The toes tend to be, obviously they show none of the constraint of confining footwear. They're very splayed, and their pads facing downward. One of the real telltale signs of a human footprint, bare footprint, is that any duration of shoe wear causes the little toe to be turned over and bent in on its side, the nail almost directed laterally. And in this case, no, the toes were very splayed, very extended, and all the pads directed downward in the plantar direction. The toes are relatively long, surprisingly long for an animal this size. You would think that the trend as exhibited in humans of shortening of the toes to resist bending stresses would have been even extended into this larger foot, which is what Dr. Krantz, my predecessor, had concluded or had inferred based on a much smaller sample size. But it's clear that the toes are actually very long. It has retained a greater degree of prehensile capability, which is afforded because of the flexibility of the midfoot and the flat archless midfoot, which spares the toes those bending stresses. The push off comes from the entire forefoot. So I'm getting a little technical, I realize, but the point is that there are, just to sum this up, there are precisely the adaptations one would expect in the foot of an enormous, heavy bipedal primate, and in sharp contrast to a simple enlargement of a human foot, which doesn't function well. I mean, one of the common maladies of individuals of excessive size or that suffer from gigantism, extreme size increase, is a breakdown of the arches of the foot and the joints of the ankles and so forth. The human foot is not designed for a body of that enormity. And so from the very beginning of the reporting of Sasquatch feet to the present, to have a foot that is so consistently distinct in exactly the ways that it should be distinct is really an impressive testimony of the credibility of this evidence.
Do you think that is it possible to forge the trace of RG? Tell us, please, about the footprints from China. You meted with Chinese forester who had footprints almost identical to those taken at Bluff Creek.
Very good. Yes. So I had the opportunity to, first let me say, as I mentioned, one of the features that is very distinctive of the Sasquatch footprint that I've been really the first person to draw attention to is this pressure ridge, and that provide, in fact, can I interrupt for just a minute and go grab a footprint cast? Okay, hold on just a second.
Отлично.
Okay, one of the examples here is a cast from the Patterson-Gimlin film site, and a distinctive feature is this pressure ridge, which unfortunately had been misinterpreted as perhaps just a tree branch buried in the sand. But it is a very distinctive feature, which I'm convinced, based on numerous lines of evidence, is a pressure ridge. In other words, the Sasquatch foot has a very flexible midfoot here, which is a pair of joints that allows for this twisting motion, a supination pronation movement of the foot. In the Sasquatch foot, because of the tremendous flexibility, the heel moves first. It moves somewhat independently of the forefoot. And as the pressure beneath the forefoot mounts, it will sometimes cause a pressure ridge, soil to be pushed back. This is a remnant of the ape adaptation of grass climbing with a divergent big toe, where the forefoot acts in prehension, gripping, and the hindfoot then in say a chimpanzee still has to move as a lever in order to elevate the body mass. On the ground, even when a chimpanzee walks, it has this flexion or break, the mid-tarsal break. So this was a very important feature that was confirmed by the footprints which I examined in 1996. And I went on to find numerous other examples. And then I had the opportunity to go to China and meet with a witness there, Mr. Yuan, and who had not only had a visual encounter, but had documented that encounter with 2 footprint casts taken at a spring where it had apparently squatted down, and as it squatted down, it bent its foot. Well, he had 2 casts to document that. They wouldn't, the producers, the filmmakers, wouldn't let me see them until they had the interview set up and the cameras rolling. But I had shown him a reprint of a paper which figured this cast very, very clearly. Although he couldn't read the English, upon seeing the cast, he was very excited and animated. And sure enough, when we opened up his case and out came his footprints, they were virtually identical, had the same pressure ridge, the same relative position and orientation, this slight angling here. And and it was just a tremendous confirmation to me that this was not only was this an accurate and real characteristic, not an artifact, but it also confirmed to me that the Chinese Yeren and potentially other examples of Sasquatch-like footprints that had been recorded, even as far to the west as the Caucasus, were in fact similar creatures. Not all relic hominoids. I mean, the Yeti, the Almas, these are different things perhaps. But in Russia, for example, the Dev of the Caucasus is a gigantic creature with a foot, whose a footprint that I could mix in with examples of Sasquatch footprints, and most people wouldn't be able to pick it out. It is so similar.
Yeah, you can hear me now, right?
Much better, yes.
Yeah. Can you dwell a little bit on the cripple foot footprints and how they are consistent with all those findings that you've had And
Exactly.
and how other footprint casts are consistent with those that you examined, which I think were Paul Freeman's footprint casts.
Right. Yes. Exactly. The cripple foot remains one of the most interesting because of the pathology or injury, one or the other, either disease or injury that it exhibits. One of the feet is twisted inward. A toe is pushed out of alignment with the others, or amputated. Could be altogether amputated. The toes are bent and enlarged. Hypertrophy of the soft tissue, and most telling is the foot is bent in such a way that on the outer edge of the foot are two prominent bumps or bunionettes, they're referred to as, which are presumably derangement of the joints where the bones are pulled apart due to inflammation and scarring and swelling and scarring around the joint capsule. So this is fascinating. My predecessor again, Dr. Krantz, had drawn attention to these. In fact, it was this case that was most convincing to him of the reality of Sasquatch. He pointed out that the bumps on the outer side were displaced, seemed to be displaced further distally down the foot than what was typical of the human foot. This simply reflects the relative position of the or the relative length of the heel segment. The bumps correspond presumably to these joints surrounding the cuboid bone. So with a human foot that has a very short heel, then the position of the bumps seems to be closer to the heel. Sasquatch, because of the midfoot flexibility and tremendous size, has a longer lever, needs a longer lever, so an increased heel segment, which then makes the lateral bumps appear to be displaced distally. So that was very, very telling. I re-examined Dr. Krantz's analysis of this, and the only difference I have is in the relative lengths of the toes. I think he was absolutely right in his assessment of the increase in length of the heel segment and its impact on the relative position of those joints around the cuboid bone. But the toes, as I said earlier, he thought that they had to be relatively short, at least as short as those in a human. And that wasn't the case. I mean, it's clear from many, many examples. I mean, even here, where the toes appear to be rather short, they look kind of like peas in a pod. First of all, they're somewhat flexed, but more importantly, as you might be able to see it with the shadow just right here, but there is a little indentation right there, and that marks a flexion crease. And just as you have a flexion crease, a very visible flexion crease across the palm of your hand that marks the position of the knuckles, that crease marks the position of the toe knuckle, if you will. So all of that is toe, not just this little bit here, but all of that. It's just that the sole pad extends up under the toes, much as the palm extends partially up under the fingers here to that degree. So with that in mind, if we look at the relative position of this pressure ridge, that corresponds to the calcaneocuboid joint. So if we look at the foot here, that's this joint right there. All right. And so the position of the pressure ridge is exactly where one would expect, given the relative elongation of the heel segment and the forward displacement of the cuboid joints. So again, it's a marvelous corroboration of the fundamental anatomy of the foot. Now, here's an interesting story about that. A distant relative of mine, a second or third cousin, went on and became an orthopedic surgeon. And we reconnected one time. He had seen me on television, and so looked me up, and I sent him a picture of the Bosberg Cripplefoot footprint casts, asked for his opinion. He was quite excited about it, found them quite interesting. And we were discussing the possible interpretations. He felt that since it was on one foot alone, the deformity, and not on both sides, then it probably was not a birth defect, a congenital defect, but rather the result of either a crushing injury or a pathology of the spinal cord, a neuropathy, which can cause those kinds of conformational changes in the foot. He suggested that given the source near a very small town, he said, was he to be presented with a patient with such a condition, he would immediately send them for a spinal cord MRI study examination. And he said, could you maybe check with some of the doctors' hospitals for someone who had this kind of deformity, who had an MRI of the spine? And I said, well, that might be possible, but you do realize that the guy would have to have 17-inch feet. and there was this pregnant pause as that fact sank in. He was so impressed with the anatomy and so engaged with its interpretation that he overlooked the actual size. So several weeks later, it turns out his boss, his supervisor, who had been out of town, was actually the specialist in the group on feet and ankles. He called me and we talked for a couple of hours about the possible diagnosis and interpretation and significance of this. He said his final words were, "I hope you don't mind, but I asked your cousin for this photo, which I now have framed. It hangs in my office, and every 3 months when I have a new set of residents to orient, this is the first case that I have them evaluate." So for orthopedists, for the medical people, there's no, I mean, it's real. The anatomy is real. They don't have the baggage of the implication that go along with recognizing or acknowledging that this could be a real thing, that such a creature actually exists. And so the anthropologists are much more much more reluctant to acknowledge that. Now, you asked about other footprints, such as Paul Freeman's. There's these several lines of evidence that all converge on this distinctive model of the Sasquatch foot. Another example, or another line of evidence for which we have examples from the Freeman collection and also other places, such as the very clear footprints that were found in Grays Harbor County, again in Washington State, but over the far western coast. In addition to exquisite full-length footprints in mud, in wet mud that extruded up around the foot and between the toes, just the outline is immaculate. There was also a series of footprints that I refer to as half tracks. And if you can imagine a track like this, but the only part that's visible is this part. Well, that I interpret as a running footprint. If you or I were to sprint down the beach because of our longitudinal arch, See, because of that arch, we have a footprint that is restricted to just the ends of the metatarsals. It's this short, it looks almost like a five-toed dog print because dogs walk up on their ends of their digits like that, digitigrade. But a Sasquatch track with that flexible midfoot, they cannot balance up on the arch. It collapses into flexion across the transverse tarsal joint with that long heel sticking up. And so when they run, they leave tracks that look like this. And so here we had a great example in Grays Harbor County. It was cast by a deputy sheriff on duty, where apparently it was investigating equipment, but then was spooked, was frightened by somebody arriving, worker arriving, and it ran back to the trees with a foot length or foot step length that doubled as it left just a series of half tracks running. Likewise, the tracks that I observed in 1996, this that we've been talking about, included several half tracks where again the the step length doubled in length as it ran, and also it left tracks that looked just like this, just a half track without where the heel never touched the ground. So all those things combined, the half tracks, the pressure ridges, the deformity with that confirm the relative lengths of the limb segments by the placement of those little bunionettes. All of those things confirm the distinctive characteristics of the Sasquatch foot and its particular segment, relative segment lengths in a remarkably convincing way. I mean, again, these are not just simple contrivances. These aren't crude, footprints left by crude wooden cutouts. They're not individuals with exceptionally, humans with exceptionally big feet. They are distinctive in many aspects of their anatomy.
So if they can be hoaxed, they should have been hoaxed by someone with a remarkable knowledge of foot anatomy, of primates and human.
Absolutely, absolutely. And we're talking about, well, for example, there are probably, now there are probably more Sasquatch enthusiasts than there are anthropologists that could explain to you the rationale for the mid-tarsal pressure ridge, namely, the mid-tarsal break, as it plays a role in our understanding of the evolution of human bipedalism. They're just, and in the 60s and 70s, there were even fewer people who had any notion of that. The study of human and fossil hominin footprints was in its infancy, and there was not a lot of comparative data. There were very few people who were studying that aspect. And so to think that somebody out of the blue, I mean, it's as silly to suggest that as it is to suggest that somebody had the skills and the knowledge to create a costume in order to, of the level necessary to create the Patterson-Gimlin film, but never used that talent in the industry, in the Hollywood industry. No other example of it exists that even touches it as far as creature effects and costume design. So I think we've done a pretty good job of identifying the blatant hoaxes when it comes to footprints. Not all footprints are so clear and detailed that all of these distinguishing characteristics are discernible. Footprints vary in quality depending on the conditions of the soil and circumstances of the trackway. And so there's quite a spectrum of those that are have no intelligible detail that upon which to base a identification, versus those that are almost a mold of the foot, a direct mold of the foot, and lots of variation in between.
Now, Dr. Meldrum, if you don't mind, let's go to the Patterson-Gimlin film. Why do you think it's still so compelling, and why does it still strong 50 years after it was filmed, actually? I've seen your wonderful analysis, Will, with Bill Muntz on the subject.
Yes.
I know that recently you've examined this film yet again with the help of artificial intelligence. So can you update us a little bit on that, on the result?
Well, just to give context, again, my forte, my expertise is in footprints, and we have an exceptional footprint record from the Patterson-Gimlin film site. Roger Patterson cast a right and a left, the clearest examples. I mean, and this was obviously fresh, having just been laid down in the sand, and it was a wet sand. The substrate, while it wasn't necessarily ideal, it wasn't like a clay, it was a very eroded shale with very angular grains that held their shape remarkably, once impressed, held the shape of the footprint very well for quite some time. This is why people could come and visit the site months later and still clearly see the line of footprints still remaining in the sandbar there. If I had just the footprint evidence from the Patterson-Gimlin film site alone, I would be quite confident in the credibility of the claim of what was on the film. But to have the footprints and the documentation of the track maker in the form of the film of the Sasquatch is all the more compelling, because then I can see the correlated anatomy, particularly the kinematics, the movement of the foot as it is actually leaving these imprints. And so, my description of how to interpret this, with the step, the elevation of the heel, the push off from the forefoot, the entire forefoot, all of that can be observed. It can be witnessed taking place as the Sasquatch walks. And then combine that with all the rest of the anatomy. I mean, I'm not just a foot specialist, I'm an anatomist, a primate comparative anatomist. And so very familiar with human form and function. And you can sit and go from the head to the toe and pick out anatomical details that one would never expect to be observable in a costume, especially, and don't forget, if it were a costume, it was a costume that was made 50 plus years ago with without the technologies or the materials that are available now. Things like four-way stretch fur were not available. You didn't have four-way stretch, you had fur cloth. It was like a carpet. You didn't have foam rubber in order to make the underlying musculature. At that time, injected foam rubber was just being experimented with in costume designs in Hollywood. John Chambers, who did the costumes for Planet of the Apes and created these facial pieces with the brows and the muzzle pieces were injected foam rubber. He got an honorary Oscar nomination or award for that pioneering effort. So it on that basis. Now, you mentioned the most recent project. We teamed up with a young computer visual effects expert, Isaac Tian, and using a technique that allowed him to superimpose multiple copies of the film. Now, the reason for this is obviously, well, not obviously, but unfortunately, the original film has apparently been lost or somehow is no longer available. So all we have are the copies This is film, not digital images. So it's film grain, not pixels. Pixels are discrete packets of information that can be replicated with very high fidelity generally. But film grain is like little crystals that are embedded in the emulsion on the film, the plastic film platen. And each batch, each copy is transferred to another slightly different pattern of crystals. So it's the potential for artifacts to be introduced with each copy is very real. So since we're dealing with copies, sometimes those artifacts can be misleading. A real famous one was the OK sign. Supposedly, as Patty's walking and her hand comes back, and instead of seeing the back of the hand, which is what's really there, people see this. I can't twist my hand that way. They see an OK sign, like it's making an OK, but on the wrong side of the hand. I mean, if you stop for a minute, it doesn't make any sense. It's simply a blemish of an artifact of that copy that was introduced and mass reproduced in the press. Well, by taking, we've successfully tracked down many copies of the film with several generations removed from the original in some cases. But by taking multiple copies of a single frame and superimposing those, the common features then are emphasized, and the artifactual features that are idiosyncratic to just individual frames are eliminated. And you end up with a frame image that is much clearer, much closer to the original than has ever been possible before. And so the resulting image, which was stabilized and created a very clear image that there wasn't necessarily the aha revelation of new information, but what it did is it did two things. It eliminated many distracting artifacts that have caused silly discussions, and two, it allowed the eye of the novice observer to focus in on the details that we were drawing attention to as comparative anatomists, and confirmed those, reaffirmed, I guess, those in our minds as well. And some of those were addressed in this recent documentary, but things like the dorsiflexion of the toes, which you'll see on that frame. I mean, you can see as the foot swings, the toes points more skyward, or dorsiflex. And that's simply the way we reach out, and we do that when we walk too. But suddenly these, peas in the pod toe, you see the entire length, because when it's swinging forward, they bend up, and the point of flexion is here, not here. So that confirms that observation very clearly, that interpretation or inference from the footprints. And more can be said about other details, the remarkable details of the facial features and proportions, the depth of the jaws, the prominence of the zygomatics, the cheeks, the small cranium, the high attachment of the trapezius. All of these anatomical details, just work your way on down through. You can see individual heads of muscles, like in the triceps. You can see the tendon of the biceps femoris behind the knee. You can, these are details that even if someone had the anatomical wherewithal, there was never any thought given to include those kinds of features in costumes in the circa 1967 whatsoever. And all you have to do is look at the costumes that were observed on the silver screen of Hollywood to see that there was nothing that came anywhere near that kind of anatomical detail.
What would you recommend to read on the subject of relic hominoid, and what are your go-to books, articles, resources, and people perhaps on the subject?
Sure. Well,
And also, I'd like to stress that we have the wherewithal to translate some articles that you might recommend to us into Russian, so they are available in Russian as well.
okay. Well, I would probably, on the subject of relic hominoids, and by that, maybe just by way of clarification, that was a term which was coined by Boris Porzhnev, a cultural anthropologist there in Russia. And his use of the term was hominoid in a more colloquial sense, that is human-like. But it also, for my purposes and my motivation for adopting that term, was that it, in a taxonomic sense, it is refers to a member of the Hominoidea, the superfamily Hominoidea, which is that which includes the human family and great apes. Well, all apes actually, greater and lesser apes, gibbon and sea among included, but that they're superfluous there. But so that it has a taxonomic as well as a colloquial sense. Relic is a term used in biology to indicate a species that, or a population that has persisted into the present from a more ancient time, and in a presumably a more restricted range, or in much more limited numbers. So relic hominoid indicates branches on our family tree, which is now seen to be extremely bushy instead of very singular. So lots of parallel lineages, some of which have persisted alongside our own until much more recently than would have been imagined 50 years ago. Even 20 years ago, we have examples that just have, The Hobbit, for example, suggests that early hominins like Homo habilis may have persisted alongside Homo sapiens until only a few tens of thousands of years ago. And so one place to go is the Relic Hominoid Inquiry, which is an online open access academic journal. It's a peer-reviewed scholarly journal, registered, with submissions from those with academic credentials or professional credentials appropriate to their disciplines, and includes original research papers, essays, book reviews, news items, lots of commentary. The idea is to get dialogue and conversation going, to provide a platform for these topics that are oftentimes shunned by mainstream academic publications. And that can be found easily through a Google search or simply going to www.isu.edu/RHI. As far as other sources, I mean, obviously there is now a plethora of publications. It's almost it's a bit of an unfortunate development, in my opinion, in this age of self-publication and so forth. It's there's a lot of very uncritically reviewed or edited materials out there. You have to be careful about what you read and what you engage with, but there are works by very respected individuals, and not to say that credentials are essential or prerequisite, but obviously that it says something about the background and experience and expertise of the individuals involved. So at the risk of promoting my own book, I would recommend Sasquatch Legend Meets Science as a starting point. I think it's a very good primer. It's now dated by a couple of decades, but it's a good starting point. Not only my own ideas, but the beauty of that production with which that publication was associated. It brought in the opinions and conclusions and observations of many different scientists. And of course, the book goes far beyond the documentary. I mean, the documentary provided the outline, but the book embellished and elaborated, I should say, elaborated upon that outline extensively. But there are others, such as, I mean, obviously, in Russia, some of the publications by Dmitry Bayanov and some of those by Igor Bortsev are certainly of historical significance. The articles by Mary Jean Kaufman about the almas, many of those have been translated into English, and your audience is not so interested in that, but they are to be found in the Relic Hominoid Inquiry in English. But the originals obviously are in Russian, are very interesting about a different, distinct relic hominoid, which sometimes gets conflated in some of the earlier literature, an effort to try to stick, I think, to a square peg into a round hole. And that is the almas tradition seems to be more akin to perhaps a relic Neanderthal or Neanderthaloid, something like Neanderthal or Denisovan, or there may be others out there to be found, those that are under the umbrella of Homo heidelbergensis, although I think we're stretching it maybe a little there, but who knows.
Are you going to make your publications available in Russia, perhaps in Russian, or what are the top 5 people you would suggest to read on the subject?
Okay. I don't have immediate plans. That hasn't really come onto my radar yet, of having my book translated into Russian. But that's something that's certainly worthy of investigating the possibilities. The top 5, I would include there. I mean, there are some of the classics which have become somewhat dated, but to really have that historical perspective, I think they're important. Obviously, one of the first introductions to relic hominoids, but under a different name, he used the acronym ABSM for short for Abominable Snowmen, was Ivan Sanderson's book. Let's see, Abominable Snowman Legend Come to Life.
I just bought the best copy on eBay. It's around $200 bucks.
Oh wow, yeah.
In the middle of February.
Yeah, but he brings a lot of materials together. It received a very unfair reception from his academic colleagues because in the 60s, the prevailing idea about human evolution was known as the single species hypothesis. The notion that the hominin niche was so particular, so singular, that there could be only one species in that niche at a time. The competitive exclusion principle of Gauss. And so human evolution was seen as a single file progression from one species giving rise to another, and so on. This notion of a bush wasn't thought to be possible. And so Sanderson, in his book was published in 1961, 62. There was no way that the scientists at the time were going to allow for a proposition of multiple subhuman species living alongside us on the globe. It just wasn't possible. So it was just written off as fantasy, as folklore, and so forth. So Sanderson's, obviously John Napier was an American, a British citizen, primatologist who was very sympathetic to the question and wrote an interesting book. Again, it's a bit dated now. It's based on very limited sample sizes. So some of his conclusions about footprints are a little bit questionable, but still from a historical perspective. Dr. Grover Krantz's book is a must read, really is. And the more contemporary ones, he's now passed, unfortunately, but Dr. John Bindernagel was a Canadian wildlife biologist. His book, North America's Great Ape, the Sasquatch, is fantastic treatment of the thesis that the anatomies and behaviors observable in Sasquatch fall right in line with what we know about existing great apes. You know, with some exceptions, some singular exceptions, such as the upright posture. But even that is a difference of degree, really, not kind. Let's see, there are others. I'm just going to add his second book, The Discovery of Sasquatch, where he basically says Sasquatch has been discovered. It just hasn't been recognized or acknowledged by science yet, and is a great philosophical treatment of how this subject has been perceived by the scientific community and the treatment that it has received. So yeah, I'm just thinking, I'm looking back at my bookshelf here. Oh, John Green. Of course, John Green is a Canadian journalist, and I mean, he really laid the groundwork. I mean, he kind of cleared the field and created opportunities. His big goal was to put this evidence before the scientific community, to kind of thrust it under their nose so they would have to address it. And so his books, his classics, On the Track of the Sasquatch, Year of the Sasquatch, Sasquatch File, not only are they great historical treatments, really give you the feel of the unfolding of this phenomenon, but he's a very insightful, very intelligent and articulate person. His book, The Apes Among Us, Sasquatch, The Apes Among Us, is a great treatment of the subject matter. And you know, it just kind of depends on what you're trying to, which aspect. Those that are scientific based. I mean, I find even some of these self-published sort of regional compendia where an individual has collected a lot of the anecdotes and stories and historical accounts from their particular region, and has made those available. Those are useful. They have their role. But as one of my as one of my professors said to me, one of my department chairs, well, after all, Jeff, these are just stories. Well, you know, it's important you get beyond the just story, that you do account for the footprints and the scat and the hair and the vocalizations and the eyewitness encounters and the photographic evidence. I mean, all those things are what lend substance to what would otherwise be easily dismissed as just stories, you know, just yarns spun by good storytellers. But that's a start anyway. But there are lots of other, as you can see in the background, though this particular bookcase is pretty much just Sasquatch, just relic hominoid subject matter. So the books keep multiplying and multiplying.
Dr. Meldrum?
Yes.
Yeah, there seems to be a problem with DNA analysis, with identifying the relic hominoid DNA. So why is it such a big problem, and why it seems that hair samples don't do much good in that respect? Right. Can you bring that Advent calendar analogy? And
yes.
Also, how much would it cost to actually establish, to actually sequence the whole mitochondrial DNA?
Yeah.
And what are the scientific advancements on perhaps eDNA and things of that?
Okay. Yeah.
Thank you.
Well, the starting point, the context for that question is the potentially very closely related relationship of of these relic hominoids to us. The nature of Sasquatch, for example, is such that, you know, we still don't know for sure. Is it an ape, something akin to Gigantopithecus, or is it something like a gigantic, robust Australopithecine, a Paranthropus that has experienced gigantism during the Pleistocene? But regardless, in either case, the potential is for a greater affinity to the human species than even the existing great apes, the chimps and gorillas, for example, of African origin. In which case, we could be talking about a difference of just a percentage or less than a percentage in nucleotide sequence. And so you mentioned the Advent calendar. I've used that analogy just to try to get an idea. Let's say, if I get my numbers straight here, this early in the morning, you know, the traditional Advent calendar with the little window leading up to the nativity, each behind each window is a little treat, you know. So, but imagine a calendar that, say, has 1,000 windows, and if this creature is only similar, or I'm sorry, is distinct from humans in only 1% of its sequences, then of this 1,000, there would be 100. Say it's less than 1%. I mean, there are some figures cited for chimpanzees that put it in the 98 to 99% range. So say this is only 0.5% or 0.1%. Say it's 0.1%, then we're down to 10 windows out of this 1,000 in the Advent calendar. Well, meaning that behind those 10 windows is a sequence of nucleotides that's distinct enough to differentiate humans from the Sasquatch. If we have a study or an opportunity, say, provided by a documentary crew to do a quick analysis, you know, they're typically going to do a mitochondrial gene, and they're just going to look at a few, maybe 1,000 base pairs. What are the odds that they have spent enough money to open more than, say, 10 of those little windows on our Advent calendar? So what are the odds in opening at random 10, because these differences are presumably scattered. They're not necessarily homogeneously scattered, evenly scattered throughout the genome, but what are the odds? They're pretty darn low. I mean, it's obviously your chances of finding one of those windows is slim, and all the resulting evidence that that study produces would point to human. Every window you opened, it would be exactly like human, indistinguishable from human. And that seems to be what we keep running into. So then combine that factor with another factor, that is that the hair that we have associated with Sasquatch, based on numerous correlations, correlated sightings and hair finds, etc., seems to consistently lack a cellular medulla, where most of the cellular remnants where DNA would be found would be located. In the absence of that, there's very little trace DNA in the keratin cortex of the hair shaft and the cuticle surrounding it. So on those rare occasions when DNA is found, it is invariably identified as human and interpreted in one of two ways. One, that it is contaminated, human DNA contaminating the sample through mishandling by the witnesses, by the collectors, or somewhere down the chain of custody. Or two, that it is simply a human hair that has been misidentified, because many of the characteristics are very similar Even the lack of a cellular medulla can be, examples can be shown amongst humans, but just not consistently. They're not, individuals with very fine blonde hair may have an acellular medulla. The third possibility that has not gotten the appropriate attention is bears on that first condition, and that is we just haven't sequenced enough to really be able to say whether it's human or not. But there's a fourth possibility now, and just recently published in the Relic Hominoid Inquiry is an interesting essay by a molecular biologist who has looked at the possibility of mitochondrial introgression, the genome introgression. Now, basically what this, and lays it out in a very accessible manner and shows examples from the literature where this phenomenon has occurred in other species. That the contact between two closely related species that has some hybridization, limited hybridization, can still result in the introgression of the mitochondrial genome. The mitochondria, the little powerhouses in the cell, have their own genome, their own DNA complement, which is distinct from the nuclear genome. And that mitochondrial genome is inherited maternally. The sperm contributes essentially nothing except the pronucleus, the nuclear DNA from the father, to the fertilized zygote. All of the cellular structures, the mitochondria, come from the mother, and then likewise are passed down from mother to daughter to so on. Well, the distinctiveness of those two genomes is such that you could potentially replace the mitochondrial genome of a species with that from another species through the hybridization, and then diffusion or introgression, the term we used, into the population, until virtually it's replaced the original. So imagine, in this case, imagine that the introgression is in the direction from the human population to the Sasquatch. In other words, the mitochondrial genome, human genome, gets introgressed and then eventually disperses until it essentially replaces what had been the Sasquatch, which presumably wouldn't be that different anyway, obviously. And we're talking about it would have no effect on the phenotype. You don't get a creature, a creature which has a Sasquatch nuclear genome and a human mitochondrial genome would still look like a Sasquatch. Now, obviously, if there was successful hybridization, there is going to be some blending of characteristics, but if there's continual back crossing into the Sasquatch population, those disappear. Those get just diluted out and disappear. And then you're left with a Sasquatch with a human mitochondria. The reason I find this so fascinating, this possibility, and it's a very real possibility, is that it would explain this confounding roadblock we seem to have, or this confusing result, which has been, unfortunately, I think, misinterpreted by some individuals, some quarters, that there is that that Sasquatch that the species itself in its entirety is the result of the crossing of human with some pre-human hominin. That's not how evolution works. You don't get an entirely new species spawned in that way. There's just not a scenario that can be drawn that makes any sense to support that. But this alternative of introgression, this passive introgression, resulting in the situation where if a legitimate sample of DNA is recovered, and only a mitochondrial, this is why we've got to get past this very superficial approach of just doing or wrote mitochondrial sequence, using some universal primer, we need to really do an extensive genomic analysis of the mitochondria and more importantly, the nuclear genome. As far as the expense, it would be very expensive and time consuming. I mean, things are continually improving. Technologies are improving, obviously, and the ability to do genomic or whole genome tests, if that would be sufficient, are becoming more attainable. You asked too about the new technologies in environmental DNA, the ability to detect trace DNA in the environment that has been shed through body secretions, body fluids, just the loss of dander, skin flaking off, remnants of skin flaking off with traces of DNA. All these things, the techniques are now available to detect even these trace amounts of DNA in soil samples, in water samples, even in air samples. That's air, not hair, but air. So that raises some interesting possibilities. Now, those techniques allow us to cast a much broader net, but we still, as far as collecting DNA samples of a rare and potentially rare and elusive species, but it doesn't address the fundamental problems we were just talking about. You're still circled back to home base, and you're confronted with, okay, now you have some DNA of a primate. How do we differentiate it from human? So it's going to require every bit, and there it may be even more problematic because it is very fragmentary DNA. I mean, it certainly would be ideal if we had a piece of flesh or a tissue, extensive enough tissue that an intact DNA sample can be the starting point. So this is a possibility. I think it's the next step because like I said, it casts the net more broadly and allows us to bring in much more by way of potential samples directly from the field without any intervening chain of custody and so forth. And those are the questions that go with that. So that's where we're at. It's been a frustrating approach, but I think I had a project in incubation just prior to the COVID pandemic, and that put things on hold, unfortunately, due to travel restrictions and field work and so on. And so as those restrictions have eased and people are kind of getting back the stock market, some of my philanthropic donors are, obviously are beholden to their portfolios, and when the stock kind of took a nosedive, the prospects became a little less optimistic. I think that has turned around a bit, and so maybe we'll be able to put together a project that will allow us to address the DNA, especially with the possibilities in mind of other ways of interpreting the results.
The drone, the Falcon project that you've been trying to launch, has it been installed or and why? And probably because of lack of finance or for some other reasons. And are you still working in that direction? And why I'm asking, we are trying to launch a similar project in Siberia, and we would also like to invite you to be part of that if you really wish to.
Right. Well, the Falcon project in its initial concept does no longer exist. It ran into both financial and technical challenges, I guess you'd say. Financially, William Barnes, who this was the brainchild of his, and it was an extremely, especially in hindsight, an extremely ambitious, definitely an overly ambitious project. I think he did his due diligence and homework and consulted with numerous engineers and so forth as far as the best strategies to pursue. I mean, at least as far as the possibilities, potential. But the practicalities were a different matter. I think the price tag of that project, as he envisioned it, was very overly ambitious, and there was there was sticker shock, as we say here. There was the BIGFOOT community never got behind it. He had, at the time I was invited to participate, he had two financial backers who essentially had committed to cover much of what he needed. And then at that time, that was right before the Great Recession here in the United States. And again, the bottom dropped out of the stock market and the real estate market. And these philanthropists that were pledging to back him had to withdraw their support because of changes in their financial status. But in addition to that, we were there were practical aspects that were very, very, very challenging as well. So anyway, the funding was not forthcoming. There was a small GoFundMe project that garnered a couple $1,000s. That money was directed into efforts to try to further this approach. Unfortunately, not directly into the field operations, but we like I said, we discovered that there wasn't the general support. I had anticipated a grassroots kind of upswell of financial support, that all these thousands of BIGFOOT enthusiasts would get out their checkbooks and make their $50 contributions. And we actually had a professional fundraising team volunteer their efforts. They were very enthusiastic about the project, said, we can do this for you. We can hit your target in a matter of weeks. And they designed and launched a GoFundMe style campaign, and were just dismayed. In 6 months, there were maybe 4 or 5 contributors, and it was just a total nosedive. I mean, it just indicated there was no support. It was not seen as practical. It was seen as overly ambitious and extravagant. And why should we pay for your toys when I could go out and buy my own toys? And so it floundered. It floundered through that period of the recession and so on. I later became involved with a researcher, and we were doing ongoing field operations. We explored the possibility of a much more conservative project, still with the lighter than air approach, to create a platform. We were working with an outfit, but a much scaled down, less expensive, less ambitious type of an airship, a drone, lighter than air drone, and a blimp. And I, spent several days down in Utah with my thumbs on the controllers, learning how to fly this thing. I mean, it still was expensive, but instead of hundreds of thousands of dollars, it was just tens of thousands. But extremely nerve-wracking to be flying this thing. I just do not have the dexterity that the younger generation raised on iPhones and gamers. We decided we would have to hire a gamer to fly this thing for us, because it was extremely nerve-wracking. We consulted with experts in thermal imagery, had custom system designed within the weight limits, et cetera, batteries, all the intricacies of trying to put this thing together. And I think that was part of the problem. The successful endeavors that are out there in, examples of wildlife surveys, there was this study where they flew the multi-copter drones and identified orangutan nests in the treetops. These were done with off-the-shelf type of equipment. But having said that, what we were trying to accomplish versus what could be done with an off-the-shelf multi-copter approach were, they were at cross purposes. The multi-copter approach, this is improving, but at the time, you could spend maybe 20 minutes with a very light payload, and that's 5 minutes out, 10 minutes, well, not even that, 7 minutes, 5 minutes back with a 3-minute buffer time to make sure the batteries didn't die and your drone come crashing to the ground or be lost out in the forest somewhere. And so there's not a lot you could accomplish. I mean, that approach is great for immediate reconnaissance, or exploring up a canyon that you're in before you hike up there physically, that kind of thing, or responding to an immediate sighting or very fresh sign and doing survey reconnaissance. You can envision all those kinds of things, but to do a protracted systematic survey with like pre-programmed grid survey pattern plots using thermal imagery. And then here's the problem: is thermal imagery has this penetrance problem, and you have to do it at night or in the wee hours of the morning. I mean, we did multiple test runs with companies and so forth, and if you put that drone up in the late afternoon or even the early evening hours, everything has that latent heat signature. You couldn't distinguish anything. But when you got up at an hour before sunup and then did it and had a test subject walk out amongst the trees, as he cleared the gaps, you could pick him up like a beacon. But as soon as he stepped underneath just the overhanging branch of a pine tree, he disappeared. So the penetrance through the canopy was very limited, which would greatly limit your ability to conduct surveys. You'd be limited to open patchy meadow forest mosaics or stream beds or lake margins, etc. But the dense forest would be essentially off limits. Someone then suggested, LiDAR may prove as a possible. This is this laser scanning technology that you've seen penetrating the dense jungle canopy to reveal the archaeological sites in Central America, for example. And it was suggested that this could eventually distinguish, large-bodied mammals, say, from the surrounding environments. Well, I did a bunch of research and digging after making some perhaps premature statements about the possibilities. I did a much deeper dive into the literature and then consulted with a couple of researchers who employ these kinds of techniques and companies that provide them. And they said, no, we're still quite a ways off from that kind of capability. It's one tool. I think that's the other direction. I think environmental DNA and as revised by the potential of a different approach to the interpretation of DNA results, sequence results, and is one prong, and the second prong is aerial survey. I mean, these are rare and elusive creatures which I think many people have an over-inflated idea of the numbers of individuals out there. I think they're extremely sparse and dispersed, and especially in areas where their habitat is experiencing degradation, and human contact is having an impact. Given that, given rare and elusive behavior, rare numbers and elusive behavior, nocturnal, far-ranging, seemingly apparently solitary or largely solitary in their foraging habits, very generalized in their diet, so there aren't exclusive resources that are constraining their movements and foraging patterns. I mean, you put all those things together, this is the epitome of the challenging species to identify in the field. I mean, talk to any person who studies rare and elusive species, like wolverines, Martin Fishers, lynx, they're problematic enough, but there's probably 100-fold those individuals than Sasquatch out there.
Dr. Meldrum.
Yes.
You must have seen the History Channel analysis of the Patterson-Gimlin film, where they took 22 or something copies of the film and examined them under an electronic microscope, I believe. And what they found was some clasping motion in her lower jaw. So what would you think that was? An animated mask or something like typical primate behavior?
I'm not sure if I'm familiar with that particular example. I assume you're not referring to the recent, it was also History Channel, The Proof Is Out There documentary that I was.
No, it's not.
Different. This is different.
Yeah.
Well, I'll have to speak to that. I can speak to that point, but not with reference to that particular example, because I'm not thinking of what it is right off the bat. But one of the things that I'm impressed with, and this is one aspect of the Patterson-Gimlin film that is, another perspective that's so impressive to me, that if you look at the film, which was shot in 1967, with 2020 hindsight, 50 plus years later, what you see is that there are aspects which in the 60s would have been assumed to be counterintuitive, like the flat face. With
how Patty anticipated the Lucy finding, right?
Right, exactly. So, one of my favorite examples to illustrate that was going to Dr. John Napier, who did write a book. He was, I think he was motivated in part because he was impressed by the film. He was amongst the panel of American or scientists in America at the Smithsonian who examined the film and rendered their opinions of it. He was probably one of the most open-minded, definitely the most open-minded. And in his book, he did come down negatively on the film, saying he thought it was a hoax, but he acknowledged, he was forthcoming enough to say he really couldn't put his finger on why. I really think part of that was, again, in the 60s, there couldn't exist such a creature in the anthropologist's mind, because that's not the way human evolution worked in their minds at that time. But what he said is, in his book, he said, but there is one point. He said, when I look at the film, from the waist up, I see an ape, essentially an ape, but from the waist down, it's very human-like in its limb proportions. He said it's almost inconceivable that such a combination of structure would exist in nature. It's a really interesting conclusion to draw. Well, it's interesting because then subsequent to that publication, which was in the early 70s, we had the discovery of Lucy and much more extensive skeletal remains of these early bipedal Australopithecines, which, you know, essentially were bipedal apes. And in the public media, they were described as, look at this, from the waist up, it looks essentially like a chimpanzee. From the waist down, it looks like a little human. Isn't it interesting how evolution has proceeded in this mosaic fashion, this unexpected combination of traits at different paces, different patterns? Well, that was the linchpin in Napier's argument for rejecting the film. And yet it actually anticipated subsequent discoveries, you know, unwittingly anticipated. And another one of those features is the flat face, but this immensely deep jaws. It had been assumed that the human profile was the reduction of the jaws and teeth, getting smaller and smaller, and being just tucked underneath this increasingly big frontal lobe of the brain, so that we ended up with a relatively flat profile. And so why would this creature with an ape-like upper body have a face that was flat? Well, we then subsequently discovered more about the robust Australopithecines that had these massive jaws, which emphasized loading the back teeth, the post-canine teeth, the molars and premolars. And in fact, the front teeth got smaller and the canines got tiny, so that the jaws could grind side to side as well as open and close. When you have these interlocking canines, see, that's about all you can do is open and close. Get rid of those and you can grind like a cow grinds, you know, to a degree. So that is a fascinating, when you take, you know, I've used an illustration where I take a robust Australopithecus like Paranthropus boisei, and you put that cranium right beside Patty, the subject of the film, and point for point, the major landmarks from the top of the head, the brow, to the cheekbones, the occlusal surface, the receding jaw, it lines up. Even the flaring gonial, the angle of the jaw here that flares outward, that creates greater attachment for these massive masseter muscles. And then, of course, the receding forehead and the massive temporals meeting up with a sagittal crest of some development to provide greater attachment for those muscles. So, you know, I can go down a half a dozen at least characteristics or combination suites of adaptations. You know, we can look at the foot, you know, we can look at the cranium and so forth. And in all those instances, in 1967, it would have cut against the grain, contradicted conventional anthropological understanding of human evolution. But now, 50 years later, we could take a picture of Patty and use it in an introductory anthro text to illustrate what a robust Australopithecine would look like if you shrank her down to 6 foot tall or 5.5 feet tall. You know, if it didn't have the stigma associated with it as the Sasquatch image, the iconic Sasquatch image, that's exactly how we envision early Australopithecines. Maybe a little less body hair, who knows? But nevertheless, that is a remarkable way. Now, again, I'm not familiar with this particular example. There has been the suggestion that you can see a gape in the mouth at one point, that it opens its mouth, or it actually looks kind of like it's you know, puffing out its lips, which is in a pout or a you know, those are typical ape expressions of intimidation or aggression. And she certainly was you know, even Roger Patterson commented that when it looked back at her, he felt like the kid playing Little League baseball when the umpire turns and, you know, it was messing around and the umpire turned and looked at him, one more time and you're out. It was that kind of an expression that he was impressed with. So, you know, it wasn't simply a static face. It wasn't, you know, so many times I get people who see this highlight that goes across the side of the nose and along the edge of the cheek, the prominence of the cheekbone, which are hairless. So they're reflecting the light, that strong sunlight. And they go, oh, look, there's a cutout. The eyes and nose are the cutout of a mask. Well, that's the most silly. Well, I shouldn't say silly. I mean, I can understand how at first glance one might think that or have that impression, but for professionals to perpetuate that, and I've seen it in publications by some of the skeptics, they point to that. The clarity is there sufficient that you can tell exactly and point to numerous examples of contemporary photography to illustrate the very same effect of that glint. I mean, this is why the soles of the feet look so bright. It's not only that the skin is lighter, but the skin is hairless. And when the foot angle comes up, I mean, just watch how the color of my palm changes from there to there. When that light comes bouncing off at the right angle, and it literally was overexposing the film just a little bit. In some places, the foot actually looks glowing. It looks puffy because the edges are burned out. They're overexposed. I mean, so many of the criticisms are so superficial and so easily corrected with a little bit of understanding of the nature of the photography and the associated anatomy.
Yeah, Dr. Meldrum.
Mm-hmm.
And just to wrap it up with a grain of humor, can you tell us about the Zaskai cave? This story with the protein bar.
Oh, yeah, that's all right.
Yeah.
Well, we went to so much of that experience. I tell you, I had such jet lag at the beginning of that trip. And of course, to their credit, Igor had arranged for a lot of activities, a lot of meetings with VIPs and dignitaries and government people and so forth, and then another red-eye flight from Moscow to Kemerovo, and I tell you, I was embarrassed because I was continually nodding off during the seminar and conference and things, but it got very real when we went out to the cave site, and I was very intrigued because of the proximity to some of the Denisovan sites and the recovery of those interesting fossils, and it became very clear as we were marching out to the cave. The path was clearly maintained. I mean, we had been told this was a remote, out of the way spot, and we'd have to trek into this remote, isolated location. And so I asked him, when it's clear, when there were bushes and limbs that were sawed and cut back, I asked him, well, who maintains this path? And Makuta was kind of indignant. Well, what are you talking about? And I said, well, there's saw marks here and there. Oh, no, we're going remote. No one goes back here. Well, I mean, there were photographs from tourists visiting the cave and the ice formations in the winter and so forth. And so I just said, well, all right. So we get to the cave, and unfortunately, I mean, it was just the footprints were so odd looking. And as I joked, there was only a right foot. There was no pattern. And in that setting, if that cave was occupied by even one individual, there would be tracks crisscrossing in and out, back and forth, and all over. I mean, there was lots of substrate there. And I made the joke, unfortunately, that what was the wild man, not Sasquatch, but was the wild man playing hopscotch, because you're only finding right feet. And of course, that made it on the television news apparently the next night. But I was a little bothered by the enthusiasm over these very contrived looking footprints and the convenient little tuft of rabbit fur that was plucked up from one of the footprints. And I started to wander back deeper into the cave with my light. And of course, a couple of the press immediately saw me taking interest, so they peel off. And before I could go a few steps, Makuta again, well, come back here, it did not want me to go back into the cave. So okay. I came back and sure enough, and then when I was being interviewed, they asked, what did I think about this? And I said, well, it seems really unlikely that this cave would be occupied with only evidence of a couple of footprints. I said, look, it's a wet cave. There's a stream running through it. There's water dripping. Creature's not going to sleep on the cold, wet floor. There would be a nest. And it was just like that was the cue. And Makuta turns his flashlight beam back into this overhang, and there's this beautiful little mattress of dry fern fronds all fluffed up into a nice pillowy mattress. No signs of it having ever been slept on. And again, I joked. I said, is there a chocolate on the pillow? And of course, then Igor immediately, before we can, even if we were going to take it seriously, take any samples or look for hair, he immediately dives in on top of the nest and sprawls out for the camera shots. So I was not having it. And so the next day, I was kind of persona non grata, and I was afraid I might not get a plane ticket home. It might be a plane ticket to Siberia, to the far Siberia, eastern Siberia. Anyway, but then that's when I thought I'd lighten it up a little bit. And well, it just kept going, because I have a very low opinion about structures, tree structures. I mean, it's one thing to have a broken tree bent over. Tree breaks are very different, in my opinion, than are structures or boweries and these types of different things that are attributed. We were hiking up to, and I can't remember the name of the overlook, but it was at a high point with a beautiful panoramic vista of the surrounding countryside. And here was a tree that had been broken off just a few feet off the ground, figure 4 break. And for a kid, it would be perfect for making a little fort underneath there, a little hideout. And of course, immediately this was interpreted as a Yeti nest, a wild man nest. And again, I was not very cooperative. And I was getting quite honestly, a little tired of the shenanigans. So in my pocket, I had a Cliff Bar, which was a protein bar, and it's like a scoop of dough, and this particular one was a chocolate almond Cliff Bar. And so I had unwrapped it and surreptitiously, it was in my hand. And so as we're talking, I'm squeezing it and massaging it into a more into the likeness of a scat sample. And so again, as was the pattern, they're doing their camera shots. Our lovely interpreters were laying out to have their photos taken in underneath this tree, and the cameras then turned to me. Well, Dr. Meldrum, what do you think? And I had Dr. Bindernagel was to my side, and I had kind of leaned over and said, John, just play along with me, follow my lead. And he goes, oh, okay. And so John was much more diplomatic. He was practicing détente, but I was getting a little impatient, and but I thought I would inject some levity. And so I said, well, look, this is a well-trodden path. I'm sure there's lots of tourists that come up to this overlook. I said, why would a wild man create, use a nest that's right here on the trail and risk being discovered? And I said, besides, look, there's sticks and rocks and pine cones. I said, any animal, even a rabbit, that has a little midden will smooth out the ground and make a nice depression to lie in comfortably. And I said, and besides, most animals that utilize a nest, a single-use nest like this, will upon getting up, they would defecate right there. They do their morning constitutional right there. I said, so we should look for, and I go, oh, and I reached down and palmed this Cliff Bar, and I said, this is what you'd look for. And when I turned it over, it made a very compelling image of a scat sample. So beside the fact that I had scooped it up barehanded, I said, but there's only one way to be sure. And so I pinched off at the end of it and popped it in my mouth and smacked my lips. And I turned to John. I says, I'm not sure. John, what do you think? And I proffered it to John, and he, without missing a beat, pinched off another piece. Oh, I think you might be right. That could be Yeti scat. Of course, Igor was not pleased by this playfulness at all, and he was glaring at me. But it was funny because our hike took much longer than had been anticipated, and so our box lunches were waiting for us down at the transport. And so it was getting late in the afternoon, and these two young interpreters come bouncing up to me, Dr. Meldrum or Jeff, they said, Jeff, do you have any more Yeti scat? We're hungry. So, like I said, I was persona non grata. It didn't go without notice that I wasn't playing along. I found it rather interesting and amusing just a few months ago, the press release where that had a picture of the commissioner there, Makuta, acknowledging what I was saying all along, that there was a bit of embellishment taking place to promote the interest in the region. I said, look at all these little ski chalets popping up like mushrooms everywhere. I said, there's a real potential for tour industry here for winter sports and so forth. I said, it's clear what the politician, the scientists may have turned their nose up at this event, but the politicians saw great potential. And there was a wild man at the gas station with a ski helmet and a snowboard. So it was clear what the overall intent was. But anyway, I think it was a great opportunity. I appreciated the opportunity, and the hospitality that was extended to us was fantastic. I mean, it was such a memorable event, and it had such great potential. Even if there was the economic motivation, I mean, that's that in some ways that's not any different than the research that I've been able to accomplish by means of the documentary productions. My experience in China, for example, would never have happened had it not been for Monster Quest and the Animal Planet. And so, I don't begrudge them that, but I draw the line at faking evidence and planting footprints and hair samples and nests and so forth. It cast into suspicion the eyewitness accounts that were shared with us, and it just poisons the well, unfortunately, if it's taken past the facilitating level to embellishing and fabrication of evidence. That was disappointing, to say the least.
Okay, so perfect story. Just Dr. Meldrum, please tell me about Todd Standing, researcher.
Yeah. Well, Todd Standing has been remains an extremely controversial figure. There's no question about that. I have been criticized for merely association, for accepting his invitation to come up at a time when I had very little basis to form an opinion one way or the other. He invited me to come up and see some of that evidence firsthand and see the locations. And so that was an opportunity I was going to take full advantage of, and had a very good experience up there with him. And I think that the things that I observed in this case were legitimate, that they were real footprints, and baits taken, vocalizations that were heard, even possibly catching a glimpse of a Sasquatch through night vision. His evidence continues to perplex me. But many of the criticisms that are leveled against them, I think, are largely baseless. And if you can't, you've got to get beyond the reaction of this isn't what I expected. That's not the basis, the proper basis for rejecting a piece of evidence. There needs to be more discriminating characteristics, or criteria for labeling something as a hoax. My attitude has just largely remained, these kinds of hoaxes, or these kinds of proffered evidences are a dime a dozen. I mean, go on the internet, there are dozens and dozens and dozens of such videos that are presented, and his are a little more extraordinary, and therefore I think warrant a little closer scrutiny. Some individuals have scrutinized him and his background and have criticized it, justifiably or otherwise. I can't say on the basis of what has been said. So again, my attitude is sort of, I don't understand when people take an overly vested interest in one individual or something like that and pursue it so doggedly, or get so upset about it. I mean, when if it involves someone else's financing or resources, I could see that, So when someone underwrites a research project to the tune of $500,000 and it's clearly hoaxed, or it's clearly misdirected at least, then that may be more of a cause for concern. In this case, I don't know that is the case. But my point is simply, the dust will settle, things will eventually be resolved, and so why expend such emotional energy about it? I've just kind of remained in the wings regarding Todd. My last contact with him was when he spoke in Mississippi. He and I both spoke at Natchez, Mississippi here recently, so it was a chance for me to kind of catch up with him and see what's transpiring. I was again disappointed. He doesn't take advantage of the opportunities he has to defend himself or present his data in a systematic way to make a conclusive argument. It's more just little teasers and then lots of discussion, lots of cheerleading, really. And a bit of hyperbole. He thinks, in my conversations with him, he thinks he's playing to the crowd, that this is what they want. There's a certain element of entertainment. And I said, no, I think you're really underestimating your audience. They want something, they want the substance. They want the beef. And you just keep teasing them with little appetizers, but you don't give them the main course. And you know, it's hard. I'm rambling because I'm really not sure how best to deal with a situation like that. I mean, he's working in an area that clearly has evidence that I've seen that impresses me, that I may have actually had my one and only visual encounter, as fleeting and as ambiguous as it was. So I'm not inclined to just throw the baby out with the bathwater, as the expression goes.
Dr. Meldrum.
Yes, sir.
What would be a message to the young zoologists and anthropologists who dare to venture into the subject of the relic hominoid and the so-called snowman and such creatures?
Well, it's interesting is, you know, with 2020 hindsight, observing my own experiences and opportunities, etc., which were quite singular. I mean, a lot of it was by chance, I think, being in the right place, the right time, with the right preparation. We're witnessing, though, I mean, but there was a lot of resistance, a lot of pushback, and you know, quite honestly, my career was in jeopardy repeatedly. I would never encourage a student to repeat the experiences that I've had, at least not without being extremely aware and eyes wide open. What I usually tell students is, let me back up. I think we're right on the cusp of a change, though, a changing of the guard. And you know, Thomas Kuhn, in his famous Structure of Scientific Revolutions, said that sometimes it takes an entire generation to pass before a new idea can take root. And I think we're witnessing that changing of the guard. There still are, you know, college deans, society presidents, journal editors, etc., that are the gatekeepers that still represent many of the older attitudes, largely influenced by this single species hypothesis paradigm that didn't allow for the possible existence of relic hominoids. You know, I thought the discovery of the Hobbit, Homo floresiensis, would change a lot of that, because, you know, here's this totally unexpected persistence of a form of hominin that you know, would have been thought to have been extinct millions of years ago and was still around just tens of thousands of years ago, if not in the present, given all the stories. I mean, even the discoverers of the Hobbit acknowledge the local traditions of the Ibu Gogo and the Orang Pendek and Dutu, and, you know, all these little people throughout Southeast Asia. So when I speak to young aspiring students, I tell them this: you can't jump into the deep end of the pool, but if you choose a profession that you have a passion for and can excel in, which is tangential in some way to this subject, be it tracking, be it wildlife biology, fiber analysis, DNA analysis, bioacoustics, you know, comparative morphology, whatever, and you become very, very good, very knowledgeable, very recognized through your publications and so forth and presentations, and you get tenure, you get some job security, which allows you a little more intellectual freedom, then from that position of authority and recognition, reputation, then turn your skills to those data that might further advance our prospects of coming to some conclusion about the question of relic hominoids. And you know, I still think we've got about 5 or 10 more years until we turn the corner. I've met numerous young PhDs, newly graduated PhDs, who are really, really interested in this subject. You know, and I give them this same advice and give them the same cautionary note. So those recent examples that I've met, it's going to take them 5 to 7 years to become established and have tenure. And then I think in that point, we'll start to see some more engagement by academics, by scientists. So I just turned 63, so I'm planning to stick around at least another 7 years in full-time academia. So I'm hoping to witness that take place, and also have the reassurance that my work won't, I mean, obviously it won't stop just because I retire from full-time a teaching position, but I'll certainly remain active and keep the fires burning. But it would be nice to have someone to pass the baton to in one form or another, just as I received it from Dr. Grover Krantz. But we'll see what you know, time will tell whether that comes to fruition or not.
Dr. Meldrum, today is January 7th, 2022. It's a Russian Christmas. So Merry Christmas to you.
Thank you.
And thank you so much for your thing which you are telling to us, because it's very interesting, fantastic. Thank you so much. And if you have possibility, let's say after half of year, for example, just if you have time, just welcome to some discussion like this.
Yes, I'd be delighted.
And because a lot of Russian people are interested in those themes, and it's very interesting. And my personal opinion, you are the best one, the number one in the world. It's my opinion. So, and thank you so much.
Well, thank you for the opportunity. It's really a pleasure, and I hope it was helpful and enlightening and amusing at times as well. So gotta make life fun.
Yeah, that's right. We just discussed with Dmitry how to do the better to make Russian version and English version, because I mean those speech.
Right.
Because it's 2 hours, it's a very so thank you so much.
Okay.
Dmitry?
Thank you very much for your time, Dr. Meldrum.
Thank you, Dmitry. Thank you.
Much appreciated. Hope we'll stay in touch.
So, nice
I
to
hope
meet
so.
you.
Please.
Personally.
Please do stay in touch.
Thank you. Bye.
Bye-bye.